Hi guys, in this video we are going to understand another important type of PCR method, reverse transcriptions PCR, in which instead of DNA the starting material is RNA. To understand RT-PCR, first you should know the PCR method which I have explained in our previous video, which I will put in the link of this video of the PCR video on the descriptions in this video. So we are using PCR to generate copies of DNA. It has unmatched applications in genetics and genomics research. Using the tag DNA polymerase, DNA synthesis occurred in conventional PCR. The tagged DNA recognizes the binding site or substrate. Here the substrate is the single strand DNA and primer binding junction, and add nucleotide which is DNTPase to growing the DNA strand. The tag has the power to amplify any type of DNA such as gDNA, plasmid DNA or viral DNA. As retrovirus and other related virus have RNA as their genetic material, their amplification and quantification are not possible using conventional tag DNA polymerase. The reason is that the tag DNA polymerase can only synthesize DNA, not RNA. During the 90s, Northern blotting was used to study the RNA, but the method was time consuming and unreliable. So, to study the RNA, we need to convert it into DNA first. After the discovery of DNA's transcriptase enzyme, the problem was solved. This DNA transcriptase enzyme is potent enough to synthesize DNA from RNA. In this video, I will cover principles of reverse transcriptions, PCR, what is procedure, what is the applications, what is the advantage and disadvantage. This RT-PCR or reverse transcriptions polymerase chain reactions is one of many variants of polymerase chain reaction. This technique is commonly used in molecular biology to detect RNA expression. This RT-PCR is often confused with real-time polymerase chain reactions, which is qPCR. However, they are separate and distinct techniques. RT-PCR is used to quantitatively detect gene expressions through creations of complementary DNA, which is cDNA, transcript from RNA. So let's see what is reverse transcription PCR. Replications, transcriptions, and translations are commonly known as central dogma of life is important to us. And this figure is the graphical representations of the central dogma process. Replications doubles DNA, transcription forms mRNA, and translations form protein or a chain of amino acid. RT-PCR measures gene expression by amplifying complementary DNA reverse transcript from RNA. We know that to study mutation or genotyping, we are using usually DNA. Then, why RNA quantification is required? Total genomic DNA has coding and non-coding sequence. The coding sequence are known as gene, which also has exon and intron. During translation, the amino acid can be formed only from coding region which form the exon. So during transcription, only the coding sequence exons of a gene forms a transcript which is or mRNA. Therefore, by quantifying the RNA, the amount of a gene present in a cell can be measured or determined. This is the whole fundamental of gene expression studies. In the reverse transcription, we know that the transcription is the process where the RNA is formed from DNA. So, in the reverse transcription, complementary DNA is formed from mRNA with the presence of one enzyme which is reverse transcriptase. RNA, set of primer, set of probes, nucleotides which is DNTPS, reverse transcriptase enzyme and PCR buffer are the key ingredients of the assay. We have to first extract RNA instead of DNA. Next, the set of primers for amplifying a gene is selected. Oligoprimers, random primers and sequence-specific primers are three types of primers commonly used here. However, which primer is applied, it depends on the type of RT-PCR assay. I already told that in the process of reverse transcription, reverse transcriptase enzyme plays an important role to convert complementary DNA from mRNA. So, reverse transcription enzymes Howard Temin and Satoshi Mizutani had discovered reverse transcriptase enzyme, although it was first isolated by David Baltimore in 1970. It is found in retroviruses and other RNA-containing viruses. In the retrovirus, the enzyme forms DNA from its RNA, which inserts into host genome at where it is replicated, transcribed and translated and forms viral proteins. The enzyme is a type of RNA-dependent DNA polymerase or RNA-directed DNA polymerase. The retrovirus has only 3 genes. There GAG genes, Pol genes, GAG or GAG genes, Pol genes and ENV genes that forms polymerase, envelope and other proteins of retrovirus. Thus the entire mechanism is known as reverse transcription. From the template RNA, the complementary DNA is synthesized using the reverse transcriptase. The process is divided into two broad steps. First, reverse transcriptions and second, amplification as well as quantification. As we said, the enzyme governs the process of complementary DNA synthesis while using probes and primer. The template is amplified and quantified. The synthesis of complementary DNA is a crucial step in the entire reverse transcriptions polymerase chain reactions protocol. The cDNA synthesis is completed into a two-step. First, strand complementary DNA or cDNA synthesis followed by second strand DNA synthesis. In the beginning, the reverse transcriptase enzyme amplifies the complementary DNA or cDNA from the single strand RNA. This is known as the first strand complementary DNA synthesis. And the reverse transcriptase enzyme does have the RNase activity that keeps the RNA from the RNase and complementary DNA hybrid into smaller fragments. Use E. coli or Escherichia coli RNAs separately into the reactions to separate the hybrid. Remember, do not use a higher concentrations of RNase H because it creates the longer RNA template prematurely. Now, the nicks generated by RNase H activity is filled by the DNA polymerase. Special type of DNA polymerase such as DNA polymerase 1 or DNA polymerase 1 and T7 DNA polymerase are used to fill the nick. The same DNA polymerase synthesizes DNA from 5' to 3' direction by removing the RNA fragments in 5' to 3' exonuclease activity. After the synthesis of the new DNA fragment, the nick between adjacent DNA are ligated by the ligase enzyme. Here use KOLiE DNA ligase instead of T4 DNA ligase, and the second strand is now ready. This is the summarized of the RT-PCR, where the RNA, we have to first extract RNA instead of DNA. Next, the set of primers for amplifying a gene is selected. It may be oligoprimers or random primers or sequence specific primer. Three types of primers commonly used here, and then it make a complementary DNA, then the PCR amplification. Depending upon that, the RT-PCR can be performed by two methods. One is one-step RT-PCR and second one is two-step RT-PCR. In one-step RT-PCR, in a single tube or single reaction, reverse transcriptions and amplification are performed. Therefore, it is named as one-step RT-PCR or one-step reverse transcription polymerase chain reaction. It is widely used in repeat quantification assays and high throughout screening due to its high accuracy, specificity, and easy to use and simple setup. In comparison to two-step PCR, two-step RT-PCR, the chance of reactions failure and contaminations are less because we are performing the reaction in a single tube here. The one-step set has several limitations because we are using various chemicals and reagents in a single reactions. Reaction conditions are sometimes compromised. Thus, it can detect less template per reactions or sample. Still, it is fast and reproducible. Also, two different reaction forms, a single sample cannot be performed because complementary DNA synthesis and amplification occurs in a single reaction. So we cannot store the complementary DNA as a stock to use in further reaction. Here we are using sequence specific primers and the reaction is single step. Thus, the chance of non-specific binding and primer dimer is too high. In the two-step RT-PCR, the reverse transcriptions and the amplification are performed in two separate reaction tubes. That is why this variation is known as two-step RT-PCR or two-step reverse transcription polymerase chains reaction. In the first reaction, reverse transcriptase enzyme nucleotide, which are DNTPS, reactions buffer or PCR buffer, oligoprimers and random primers are used to build a complementary DNA. After completions of the reactions, we get complementary DNA stock, which we can store to use in the future, or we can do amplification for the gene expression study. In the second reaction, which all other PCR reagents, the normal tag DNA polymerase conducts are reactions for quantification. Here, reverse transcriptase is not used. In second reaction, complementary DNA is amplified and quantified, and one of the biggest advantage of the two-step method is that we can store the complementary reverse transcript from the RNA. In two-step RT-PCR, we are using three sets of primers, one set of oligoprimers, one set of sequence-specific primers, henceforth, and it is highly specific compared to the one-step method. The chances of the contaminations or reactions failure is higher in the two-step RT-PCR reaction. Usually, it is used in viral gene expression studies. And the picture shows four different priming method for the reverse transcription step in two-step assay of reverse transcription polymerase chain reaction. If we summarized the process of the RT-PCR, that is sample preparations, next selections of primers, third step is reactions preparations, and next one RT-PCR cycle conditions, and the last step is standard synthesis. Many clinically important viruses have genomes composed of RNA. RT-PCR is useful for detecting such viruses. RT-PCR is being used for the detection of the following viruses, that is dengue viruses, hantavirus, human metapneumovirus, severe acute respiratory syndromes, or SARS virus. Now, RT-PCR is famous for to diagnose the COVID-19 or coronavirus detection. The whole mechanism of process is same as RT-PCR. Here, the individual is positive or negative. How would we know that one person is positive or negative? By using the method of RT-PCR. A reverse transcription polymerase chain reaction assay was developed to rapidly detect the several acute respiratory syndrome associated coronavirus, which is SARS-CoV virus. The assay based on multiple primer and probe set located in different regions of SARS-CoV genome. The technique is swab is used to take of the virus from the back of the nose or throat, which respectively called nasopharyngeal swab and oropharyngeal swab. The swab is put in vial containing a buffer solution which preserves the virus till the time the sample is processed. And the genetic material in the sample, viral, bacterial and human, is extracted and purified using buffer solution. The RNA viral DNA is converted into DNA using an enzyme, which I told in the previous. The name of the enzyme is reverse transcriptase, which includes the process of reverse transcription. Then the PCR amplification is going on. And primers have fluorescent probes or dyes which are released other is cycle of replication. Specialized equipment is used to determine the presence or absence of virus in the sample using this dye. You are positive or negative, how would you know? After is a graph seeing the x-axis is indicates time and the y-axis indicates the fluorescence. If the in the graph there is change in the fluorescence by times, this shows the positive, or the person is affected by coronavirus or COVID positive. If there is no change in graph, its time and the fluorescence, so the person is not affected by coronavirus or is the negative. RT-PCR may also be used to detect other microorganisms like bacteria, parasites and fungi by targeting their rRNA. Detecting of mRNA using RT-PCR helps to study the gene expressions of both microorganisms and human host cell. The advantages of the essay of the method of RT-PCR. The method can do quantitative as well as qualitative analysis. It requires a small amount of sample gene expression studies. Post-PCR processing such as agar-rose gel electrophoresis is not needed here. The method is simple, easy to use, rapid and cost effective. And the specificity and the sensitivity of the RT-PCR is enormous. And the disadvantage, it has also disadvantage. The method is extremely sensitive. Even a small amount of DNA contaminations can lead to false result. The method is restricted for some assays as higher expertise and experimentations are required to develop new assays. So in this topic, in this video, I discuss about the reverse transcription polymerase chain reaction. This RT-PCR process is now very popular to detect, to diagnose a disease which is a pandemic disease, COVID-19. In this video, I discuss about the principle, the procedure, the application and the advantage and disadvantage of the RT-PCR reverse transcription polymerase chain reactions. If you like this video, please share with your friends and don't forget to subscribe or don't forget to press the bell icon, because if you will get more video like this, you have to press the bell icon. Thank you from Knowledge of Life A2.